Welcome to Odisha Net Academy. Dear students, now we are going to discuss about protein-protein interaction, the methods which are discussed which are involved in the protein-protein interaction study. So in the last class, we have discussed about density gradient centrifugation. Okay, total principle and how with the help of density gradient centrifugation we can detect protein-protein interaction. Already we have discussed in our last class. Today we are going to discuss about immunoprecipitation reaction, cross-link and non-cross-link. With the help of this reaction we can detect whether two proteins are interacting or not interacting. So first you should know about immunocomplex and immunoprecipitation. So when antigen and antibody, okay, antigen and antibody form complex, it is called immunocomplex. Okay, so suppose this is the antibody and the antigen get attached to its corresponding antibody to form immunocomplex. Normally that immunocomplex is a little bit higher molecular weight, higher shape and size than that of free antibody, than that of free antigen. That's why with the help of centrifugation force, it is easily, that complex is easily pellet down. The pelleting down of immunocomplex by applying centrifugal force is called immunoprecipitation reaction. So in this case you have to separate the immunocomplex, okay, from the unattached molecules like free antibody, like free antigen, like that. And by using this principle we can detect whether protein A and B are interacting or not interacting. Okay, so now come to stepwise discussion. First take beads. So it is a polysaccharide, polysaccharide. You have to first take beads, then add antibody against any one of the protein, antibody against A or antibody against B. Suppose in this case I have taken antibody against what? A. So now what happens? The antibody get attached to beads, which is polysaccharide. Okay, so with the help of non-covalent bond. So this is called non-cross-link. This is called non-cross-link type of immunoprecipitation. So with the help of non-covalent bond or weak interaction, antibody get attached to beads. Now in this case or after that step, you have to add A and B incubated solution. Okay, so in one container, that is what happen drop, you have to add A solution and B solution and incubate. After that you have to add that AB solution to this container. Now what happen? First say what happens to the, or which protein is attaching to antibody irrespective of AB interaction. As I have taken antibody against A, definitely A will come and get attached to here. Okay, whether A interact with B or not, that does not matter. Okay, but always A come and get attached to this antibody because we have taken antibody against A solution, A protein. Suppose, okay, A and B are interacting. Case 1, A and B are interacting. So what happened? Now along with A, who will come and get attached? B. So the complex is made up of bead, antibody, A protein and B protein. Got the idea? But actually, as I assume A and B are interacting, then I can say A and B are present or attached to antibody. But actually I have no idea. I have to find out that one. That's why you have to centrifuge. So only immunocomplex will come to palette, and which are unbound, non-bound, that are coming to supernatant. So palette now contain, in interaction case, palette now contain bead, then antibody, then A protein, then B protein. Okay, so now I have no idea about which protein get attached. So that's why to know that one, you have to now separate all the protein from each other. No need of now interaction because already the interacted thing come to palette, get separated. So with the help of beta mercury ethanol and HGS, so we have to separate these proteins and run through HDS page. Run through HDS page. Okay, now what happened? So if you are getting A band as well as B band in the gel, definitely A and B are interacting. Along with other two band you can get, because due to the actions of HDS and beta mercaptoethanol, so beta mercaptoethanol digests the disulfide linkage present in between heavy chain and light chain, so making them separate. That's why you can get heavy chain and light chain. These are the protein of antibody. So ignore that one, but focus on A and B band. So if in palette side, if you are getting A band and B band, then definitely you can say A and B are interacting. And you can cross check your result by treating the supernatant and running through that what HDS page, same treatment, and you are getting no band. No band means, okay, there is no free A, there is no free B. Okay, and all A and B are getting what attached to each other and come to palette. So that's why it also provide or support the result of palette side, means A and B are interacting. Similarly, if A and B are not interacting, then what happens? Now, even if A and B are not interacting, still A get attached to antibody, because I have taken antibody against A. And B come to supernatant after centrifugation. After centrifugation, B come to supernatant. So after treatment of what? Palette with the help of beta mercaptoethanol and HDS, you are getting three band. One is heavy chain, one is for heavy chain, one is for light chain. It's okay. And only what? One band for protein A. And there is no band for protein B. Okay, as and we are not interacting, so B comes to supernatant. So test that one, check that one. Yes, now I am getting B band in supernatant side. So it indicates that A and B are not interacting. Got the idea? With the help of immunoprecipitation method, with the help of this simple technique, that too non-crossing wala immunoprecipitation, you can detect whether two proteins are interacting or not interacting. So one problem is there. Problem is, so during as I told you, this is the non-covalent bond. Attachment is weak interaction. So there is a chance of detachment during the centrifugal force action. So that's why what you have to do now, another what small modification you can do, that is cross-link. So you have to cross-link the bead and antibody permanently with the help of cross-linker. Normally we are using glutaldehyde. Okay, we are using glutaldehyde as cross-linker. You can use formaldehyde also. So and rest steps are exactly same. Okay, so what happens? So in case of interacting case, what happens? You can get A band and B band, but you may not get heavy chain, because heavy chain permanently attaches to bead, remain in the what? Well, it cannot enter into gel. Okay, that's why instead of getting heavy chain, okay, you can get A chain, B chain and light chain. And here no band. It indicates that A and B are interacting. Similarly, in case of what non-interacting case, what happens? So in palette side, what happens? Only A protein is present. So you can get band for A protein, band for light chain, but no band for heavy chain, because heavy chain still attaches to that beads. And supernatant is coming to palette, and you are getting what? Band for B. Okay, so B is coming to palette. Your that is supernatant. B protein is coming to supernatant. So now you are getting B band in supernatant HDS page. So it indicates that A and B are not interacting. Now got the idea? So with the help of this another simple technique, one we have discussed about density gradient centrifugation, and this one the second method, okay, through what you can detect whether two proteins are interacting or not. So it is a little bit more advanced or more accurate than that of previous density gradient centrifugation. Why? Now here we are using protein and their what? Antibody. So protein and antibody or antigen antibody interactions are very specific. So chances of non-specific interactions are very less. Got the idea? So this is the one another method. Okay, so in the next class we may discuss what other advanced techniques to detect protein interaction. Okay, so thank you everybody.